Hello everyone, I am Mrs. S. Sumati, working as Assistant Professor in the Department of Botany in St. Josephs College of Arts and Science for Women, Ozur. In this video, we are going to see the tissue systems in plant anatomy. Plant anatomy, the study of internal structure and organization of plant is called plant anatomy. In Greek, ana means as under, teminen means to cut. Father of plant anatomy, Nemei grew. Plant anatomy or phytotomy is the general term for the study of the internal structure of plants. What is meant by tissue? A tissue is a group of cells that are alike in origin, structure, and function. The study of tissue is called histology. A plant is made up of different types of tissues. There are two principal groups, one meristematic tissues, two permanent tissues. A meristem is the tissue in most plants containing undifferentiated cells called meristematic cells found in zones of the plant where growth can take place. Characteristics and classification of meristematic tissue. The term meristem is coined by C. Nageli. The meristematic cells are isodiametric and they may be oval, spherical, or polygonal in shape. They have generally dense cytoplasm with prominent nucleus. Generally, the vacuoles in them are either small or absent. Their cell wall is thin, elastic, and essentially made up of cellulose. These are most actively dividing cells. Meristematic cells are self perpetuating. Based on position, meristematic cells divided into three groups. 1. Apical meristem. Present in apexes of root and shoot. It is responsible for increase in the length of the plant. It is called as primary growth. 2. Intercalary meristem. Occurs between the mature tissues. It is responsible for elongation of internodes. 3. Lateral meristem. Occurs along the longitudinal axis of stem and root. It is responsible for secondary tissues and thickening of stem and root. Example, vascular cambium and cork cambium. This is shoot apical meristem which shows leaf primordium, procambium, protoderm, ground meristem and vascular tissues. This is root apical meristem which shows root cap, apical meristem region, zone of cell division, zone of elongation, zone of maturation and root hairs. This is intercalary meristem which shows elongation of internodal region. This is lateral meristem which shows secondary thickening in root and stem. Based on origin, meristematic tissues divided into two types. 1. Primary meristem. It is derived from embryonic stages and differentiated into primary permanent tissues. 2. Secondary meristem. It is derived during later stage of development of the plant body. It produces cork cambium and interfascicular cambium. Based on function, meristematic tissues divided into three groups. 1. Protoderm. It gives rise to epidermal tissue system and develops into epidermis, stomata and hairs. 2. Procambium. It gives rise to primary vascular tissues. Example, xylem and phloem. 3. Ground meristem. It gives rise to all tissues except epidermis and vascular strands. Based on plan of division, meristematic cells divided into three groups. 1. Mass meristem. It divides in all planes. Example, endosperm, young embryo and sporangium. 2. Rib meristem or file meristem. It divides anticlinally in one plane. Example, development of cortex and pith. 3. Plate meristem. It divides anticlinally in two planes. Example, development of epidermis. Permanent tissue. The permanent tissues develop from apical meristem. They lose the power of cell division either permanently or temporarily. They are classified into two types: one, simple permanent tissues; two, complex permanent tissues. Simple permanent tissues. Simple tissues are composed of one type of cells only. The cells are structurally and functionally similar. It is of three types: one, parenchyma; two, collenchyma; three, sclerenchyma. Parenchyma. Parenchyma is generally present in all organs of the plant. It forms the ground tissue in a plant. Parenchyma is a living tissue and made up of thin walled cells. The cell wall is made up of cellulose. Parenchyma cells may be oval, polyhedral, cylindrical, irregular, elongated, or armed. Parenchyma may store various types of materials like water, air, ergastic substances. It is usually colorless. The turgid parenchyma cells help in giving rigidity to the plant body. Partial conduction of water is also maintained through parenchymatous cells. Types of parenchyma. 1. Aerenchyma. Parenchyma which contains air in its intercellular spaces. It helps in aeration and buoyancy. Example, nymphea and hydrilla. 2. Storage parenchyma. Parenchyma stores food materials. Example, root and stem tubers. 3. Stellate parenchyma. Star-shaped parenchyma. Example, petioles of banana and canna. 4. Chlorenchyma. Parenchyma cells with chlorophyll. Function is photosynthesis. Example, mesophyll of leaves. 5. Prosenchyma. Parenchyma cells became elongated, pointed and slightly thickened walls. It provides mechanical support. Here we can see the different types of parenchyma tissues such as chlorenchyma, aerenchyma and storage parenchyma in bean. Collenchyma. Collenchyma is a simple living mechanical tissue. Collenchyma generally occurs in hypodermis of dicot stem. It is absent in the roots and also occurs in petioles and pedicels. The cells are elongated and appear polygonal in cross section. The cell wall is unevenly thickened. It contains more of hemicellulose and pectin besides cellulose. It provides mechanical support and elasticity to the growing parts of the plant. Types of collenchyma. 1. Angular collenchyma. It is the most common type of collenchyma with irregular arrangement and thickening at the angles where cells meet. Example, hypodermis of datura and nicotiana. 2. Lacunar collenchyma. The collenchyma cells are irregularly arranged. Cell wall is thickening on the walls bordering intercellular spaces. Example, hypodermis of hypomia. 3. Lamellar collenchyma. The collenchyma cells are arranged compactly in layers or rows. The cell wall is thickening is at tangential walls. These thickening appear as successive tangential layers. Example, hypodermis of Elianthus. Here we can see the different types of collenchyma tissues such as angular collenchyma, lamellar collenchyma and lacunate collenchyma. Sclerenchyma. The sclerenchyma is a dead cell and lacks protoplasm. The cells are long or short, narrow, thick-walled and lignified secondary walls. The cell walls of these cells are uniformly and strongly thickened. The sclerenchymatous cells are of two types: one, sclerites; two, fibers. Here we can see the different types of sclerenchymatous cells such as sclerites and fibers. Difference between sclerites and fibers. Sclerites are short cells, usually short and broad, occurs individually or in small groups, may be branched, develops from secondary sclerosis parenchyma cells, and fibers, long cells, narrow, elongated pointed ends, occurs in bundles, commonly unbranched, derived directly from meristematic tissues. Here we can see the longitudinal and cross sections of parenchyma, collenchyma, and sclerenchymatous tissues. In this video, we had seen meristematic tissue and simple permanent tissues. Thank you.